Good afternoon, everybody. I put this slide in to remind me and to remind the audience that even though this afternoon's session deals with heat, ultraviolet radiation, while we think of it in the context of overall solar radiation in many cases, really crosses boundaries between heat, cold, altitude, space flight. So it's ubiquitous in environmental extremes for the most part. What I'm going to try to do today is to use solar UV radiation as a theme for kind of molding some physiological studies that we've done that have to do with vascular health, with some anthropology studies that very good colleagues of mine at Penn State have done that have to do with the evolution of skin color and pigmentation. And so the outline of my talk, I'm going to talk about UV radiation exposure in the skin. I'll talk specifically in the context of UVR risk for athletes and committed exercisers. I'll then switch gears and talk a little bit about the impact of radiation on skin vascular health, which has been a big interest of ours. I think everybody's very familiar with cancer-causing aspects of some forms of UV radiation, but the National Cancer Institute, which is part of the National Institutes of Health in the United States, have now opened an entire portfolio of funding having to do with non-cancer effects of solar radiation on the skin. And then finally, I'll talk about radiation and the evolution of skin pigmentation, focus on the vitamin D-folate hypothesis. And in between, I'll talk a little bit about sunscreen, and I'll hopefully talk and get you thinking about two recent controversies having to do with UV radiation and human health. So just as a background and to remind everybody that there are three forms of UV radiation delineated by wavelength. The longer wavelengths are UVA, middle UVB, shorter wavelengths, UVC. UVC radiation essentially bounces off the atmosphere, doesn't create much of a health risk for humans. But UVB radiation penetrates the skin, but not beyond the epidermis. And so UV radiation is most often associated with sunburn. UVA radiation does penetrate to deeper skin layers and is associated in most contexts with photoaging and wrinkles and aging of the skin in general. But importantly, even though UVB radiation doesn't penetrate beyond the epidermis, both UVA and UVB radiation produce inflammatory responses, inflammatory cytokines and increases in reactive oxygen species within those deeper dermal layers of skin tissue. So this study came out a few years ago. It's a very, very often cited study. Those of you who work in exercise physiology or sports medicine will appreciate it. And the fact that being active, having leisure time physical activity, is associated with decreased risk and decreased prevalence of a large number of types of cancers, from esophageal, gallbladder, liver, lung, and so on. For all of these cancers, the hazard ratio is less than 1 if you're physically active on a regular basis. The exception, of course, is malignant melanoma and other types of skin cancers, where if you're physically active outdoors, the risk of skin cancer actually increases. The only other one that has a similar type of effect is prostate cancer, and there are some odd reasons for why that occurs. So there is a lot of confusion in the literature about personal doses and personal exposure to UV radiation in exercise and sport, and that's because the field flips back and forth between different units of measure. So for example, the MED stands for multiples of minimal arrhythmia dose. So 1 MED is the minimal dose necessary to cause a visible skin reddening within 24 hours. And the problem with that, it has its place in the literature, but it's very variable and depends, of course, on skin pigmentation. So a lower MED is going to cause erythema in lighter skinned people than in darker skinned people. And skin color is most often categorized according to a well-known scale, the Fitzpatrick scale, which has skin types of 1 to 6, actually 1 to 7, where 1 are very lightly pigmented or freckled skin and almost always burns, to very dark skin that in the old context of the Fitzpatrick scale said never burns, but we know now that that's not necessarily the case. So MED depends on pigmentation. To get away from that and to standardize doses of exposure to UV radiation, most scientists in this area use SED, or standard erythema dose. So 1 SED is 100 joules per meter squared, and that eliminates the individual skin differences in the response and standardizes the exposure. So if that were not clear enough, the public health world uses the UV index, and that's the one I think most people in this room would be familiar with. So the UV index is a time-weighted average where 1 UV index hour is about 1 SED. And the UV scale goes from 0, which is low risk if you're unprotected, to above 11, which is extreme risk. And just to put it into context for you, 1 hour of sun exposure at a UV index of 5 is likely to elicit a skin reddening response in skin types 1 to 3 and maybe 4. And the literature quickly goes back and forth among these different dose types of responses. In terms of how much sun exposure people should get when they're unprotected, the ICNIRP guideline is that the maximum biologically effective UVR over an 8-hour period, and this was designed for industry and the workforce, should be limited to a dose equivalent to approximately 1 to 1.3 SEDs. So let's put that into context for athletes and regular exercisers. There are a lot of studies out there, epidemiological studies, looking at athletes and committed exercisers, showing that in Tour de Suisse cyclists, Ironman triathletes of the World Championships in Kona, alpine ski instructors, and so on, they average 6, 10, even higher MEDs. And in larger samples of hikers and tennis players and runners, the average is even higher than that. And what that means is that all of those regular exercisers and outdoor exercisers exceed the recommended exposure limit by up to 8-fold, and often reach that limit in less than 20 minutes. And these are exercisers and athletes that in some cases train 4 or 5 hours a day, every day. So the exposure risk for athletes and regular exercisers is high. So we've been interested, as I said, in non-cancer aspects of UV radiation. Specifically, what does UV radiation do acutely and chronically, I'm going to stick to acutely today, on skin vascular health, the small blood vessels in the skin that serve a nutrient function in skin health, but also serve a thermoregulatory function in terms of the ability to vasodilate and vasoconstrict. You saw something this morning in Anthony's talk that looked very similar to this, in that one of the highlights of healthy vascular function is a preponderance or availability of NO to support cutaneous vasodilation and vascular health. And NO is derived from L-arginine with the coupled enzyme nitric oxide synthase. And a couple important features of this are that for this enzyme to remain coupled, this cofactor, BH4, or tetrahydrobiopterin, has to be present in abundance. And, as you heard this morning, uncoupled NOS can lead to generation of superoxides, which then couple with nitric oxide to form peroxynitrate, so this high oxidant environment. In order for this to occur in a healthy fashion, One of the things that stabilizes nitric oxide synthase and allows us to form NO is the bioactive metabolite of folic acid, or 5-methyltetrahydrofolate. So folate not only helps BH2 form BH4 to support NOS, but also serves as an antioxidant to quench reactive oxygen species. So folate plays a very important role in skin vascular health. We use a technique in our laboratory that some of you are familiar with called intradermal microdialysis. And using this technique, we put a very small, fine catheter under the skin surface. It's got a semi-permeable membrane, and we can infuse a variety of drugs into that membrane to allow them to interact with capillary beds so that we can block metabolites, we can support various aspects of the metabolic pathways, we can put antioxidants in. And over that site where we have the semi-permeable membrane, we measure skin blood flow with a laser Doppler probe. And in many cases, we locally heat that area as a stimulus to vasodilate this area. And this is what 4 of those probes and fibers look like on the forearm of a subject in the lab. This subject is wearing a water-perfused suit for a whole body heating study. The studies I'm going to talk to you about today really involve just local heating of that small area of skin. So Tony Wolfe, who's a PhD student of mine, published a paper recently that looked at the effect of acute ultraviolet radiation on that NO-mediated vasodilation in human skin. And so in these experiments, what we do is we locally heat the skin. This is cutaneous vascular conductance. So when we locally heat the skin, you get a rise in skin blood flow. You reach a small peak and a nadir, which is due to an axon reflex response. And then it starts to increase and reaches a local heating plateau that's primarily nitric oxide dependent. We then infuse through those fibers L-name, an NO synthase blocker, to decrease to a new plateau. And the difference between this plateau and this plateau is the percent of the blood flow response that's mediated by nitric oxide. And then at the end, we perfuse sodium nitroprusside to get a maximal dilation. In this study, what you're seeing here is one arm that has been exposed to 75 seconds of UV radiation before this response occurred, and the other that was a non-exposed arm. And so we did this in a number of subjects. I'll point out that number one, they're young, healthy subjects, and they have fairly light to moderate skin melanization. And when you do that, the percent NO contribution, even after 75 seconds of UV radiation, and this is all UVB radiation in this study, by the way, caused a significant reduction in the percent of the response that is responsive to NO. And I'll ask you to maybe suspend disbelief and think of that as healthy vascular function when you can have a larger NO-mediated response. In other fibers in that same arm, we infuse the general antioxidant ascorbate. And what happens now is if you put in ascorbate and you quench those superoxide radicals, you correct the response. In another one, we put in that folate analog, 5-MTHF, and the same thing happens. So what happens here is if you block this response directly, you get increased NO production and vascular health, and UV radiation blocks the 5-MTHF or decreases it. Both UVA and UVB radiation serve to block this beneficial pathway of folate. And so the response, instead of vascular health, is then vascular dysfunction. We repeated those studies in the presence of sunscreen, kind of a generic SPF 50 sunscreen on the arm before we started local heating, and in the presence of simulated sweat. So these are sprayed water droplets that have the same electrolyte composition as human sweat, but keep in mind that they're not coming through the sweat glands and potentially having effects on the circumpore cells of the sweat glands. They're just sitting on the skin. We repeated those experiments, and here's again the control situation where this is the non-radiated arm, this is the radiated arm, with the exception of this one crazy outlier that everybody has in their studies. UV radiation decreases NO-dependent dilation, but that's essentially reversed by the presence of sunscreen or simulated sweat. So sunscreen good, radiation bad. So controversy number one. You may have seen this in JAMA a couple months ago. A study came out in which they took a total of 24 men and women, young, healthy men and women, and they put on 4 different sunscreen in a cross-sectional design. Two were sprays, one was a lotion, one was a cream. These are all chemically based sunscreen. They're chemically based sunscreen. There are also barrier sunscreens like zinc oxide and things that reflect the UV radiations. Chemically based sunscreens are designed to absorb the UV radiation. When they did that, what they showed was that in all 4 cases, all 4 forms of sunscreen, they were able to measure very small quantities of substances within the sunscreen in the plasma. And so the scientific world kind of went wild that systemic absorption of sunscreen ingredients all exceeded the FDA levels for avobenzone in the plasma, and the dermatology world kind of went wild. We're no longer prescribing chemical sunscreens for any of our people, only barrier sunscreens. But if you delve deeper into this paper, the controversy is these are incredibly small quantities of a substance that we don't even know is harmful. And so the conclusion in the paper, which is overlooked by the overexcited dermatology world, is that this supports the need for further clinical significance of these findings, because at this point in time, there really are none. But it's a current controversy. You'll see more about it. So we have this situation where UV radiation hits the skin. I've described so far what you see in this red box, which is the negative effects of UVB and broad spectrum UV radiation working through folate metabolites to reduce nitric oxide bioavailability and decrease skin vascular health. The other side of the coin, however, is the fact that in particular UV radiation is absorbed in the skin and results in the formation of vitamin D. So there's an increased vitamin D that increases NO bioavailability and augments NO-mediated dilation. So you've got 2 sides of the same coin. The folate side that says that UV radiation is bad on the skin, the vitamin D side that says UV radiation is good on the skin. So I'm lucky to work with one of the world's foremost biological anthropologists, a woman named Nina Jablonski, and her husband, George Chaplin. If you ever get the chance, Nina has a fantastic TED Talk that you really should listen to about skin and skin pigmentation, and has written several best-selling books on the topic. If you look at the world in terms of thermoclines of annual UVB radiation, you can see that the annual mean preponderance of UV radiation is close to the equator and decreases the further you go from the equator. But the coefficient of variation for UVB radiation is just the opposite. It becomes much more sporadic in these locations and much more constant close to the equator. And so Nina has written, as others have, that human skin pigmentation, the amount of melanin in your skin, evolved as the product of two clines produced by natural selection, specifically to adjust pigmentation to UV radiation. This one cline of darker skin is generated by high UV radiation near the equator, so that people who live near the equator evolved with dark, photoprotective, melanin-rich, folate protective UV radiation, whereas the others produced for UVB photons to sustain synthesis of vitamin D in low UVB environments. And that resulted in the evolution of lightly pigmented skin. So when you look at why we have different skin pigmentations, it has nothing to do with a thermoregulatory advantage, or very little to do. It has little to do with skin reflectance, or preventing sunburning, or preventing cancer. The major causative agent in the evolution of skin pigmentation is that balance between folate and vitamin D metabolism, folate degradation and vitamin D metabolism. So UV radiation promotes vitamin D synthesis and folate degradation, and it's always a balancing act. Darkly pigmented skin protects against folate degradation. Lightly pigmented skin allows for lots of vitamin D to be produced in the skin. So the vitamin D folate hypothesis proposes that those two evolved as a balancing mechanism, that skin pigmentation evolved to balance those two effects. So again, increased melanin blocks or decreases this effect and this effect. So it's beneficial with respect to folate degradation. But it also blocks this positive effect of the pathway that produces vitamin D and augments skin health and skin vasodilation. And that would have been okay if people hadn't started to migrate. But what happened as a consequence of migration patterns, many darkly pigmented individuals now reside in areas of low UV radiation and variable UV radiation, and individuals with lighter pigmentation are exposed to heightened levels of UV radiation in climates closer to the equator. So these individuals are now at risk of negative UVR adaptive responses because of an increased risk of not enough vitamin D if you have darker skin and live away from the equator, and an increased risk of folate breakdown if you have lighter skin and you live closer to the equator. Why is that important? It's important for cardiovascular health in general. African American populations have higher levels of hypertension, higher incidence of cardiovascular disease, earlier onsets of cardiovascular disease, and higher morbidity and mortality rates for cardiovascular disease. And one of the leading hypotheses for why that occurs in darkly pigmented skin, and again, I'm not overlooking socioeconomic factors and access to health care and those sorts of things, but from a purely physiological standpoint, one of those proposed roles is that the low ability to synthesize vitamin D in the skin is responsible for the genesis and maintenance of elevated blood pressure in African American populations. We're studying this, Matt Brothers and other people have been studying this, and it really looks like it's easy to document, at least at the microvascular level in terms of vascular health. Controversy 2 is sunscreen the new margarine? So there have been a spate of recent reports in the popular literature, and one of the big proponents of this idea is a dermatologist here in England named Richard Weller, talking about how sunscreen is bad because sun exposure is inherently good. And here's the reasoning. The rates of high blood pressure, heart disease, stroke, and overall mortality all rise the further you get from the equator. If you give people vitamin D supplementation, it doesn't decrease their cardiovascular risk. So it may be the sun itself and not vitamin D, according to this hypothesis. For every person who dies of skin cancer, more than 100 die from cardiovascular disease. Stay with me. Sun worshipers have a higher incidence of melanoma, but are 8 times less likely to die from it than people who don't get skin cancer from sun exposure. And so the conclusion of all of this, and this is just one of many reports that you can find in the literature, and there's a big push with some really good scientists behind it that say avoidance of sun exposure is a risk factor of a similar magnitude as smoking in terms of life expectancy. So if you always stay out of the sun, you always slather on lots of sunscreen, you avoid the sun at all costs, you're just as likely to die from cancer as you are as a smoker. I said it was a controversy. I didn't say I had the answer. So, in summary, UVR risk is very high for athletes, committed exercisers, people who spend a lot of time outdoors. UVR exposure has a variety of non-cancer effects on the skin, including those on skin vascular health and cardiovascular function. UV radiation promotes vitamin D synthesis and folate degradation. And so there is a need for some sun exposure. Some people have said that that amount of unprotected sun exposure is the equivalent of about 20 minutes in the noontime sun as the maximum for vitamin D metabolism. And individuals with darkly pigmented skin are susceptible to hypovitaminosis D and potentially then evolving into other types of cardiovascular disease because decreased vascular function leads to atherosclerosis, which is kind of the building block of all forms of cardiovascular disease. And finally, sunscreen beneficially mitigates the impact of UVR not only on cancers, but on skin vascular health. So I'll stop there and happy to answer any questions.